Good morning, everyone. So we are talking about the biological classification. So in the biological classification, so we had discussed about kingdom Monera, and we are discussing about the kingdom Protista. In the kingdom Protista, we have completed euglenoids, dinoflagellates, chrysophytes. Now let us start with the slime molds. So slime molds. Here, slime molds are saprophytic protistans. They are the saprophytic protistans. So in the slime molds, there is no cell wall. So cell wall is absent. Cell wall is absent in the slime molds. Instead of cell wall, spore wall is present. Instead of cell wall, spore wall is present in the slime molds. Okay. So here, the body of the slime molds move along decaying twigs and leaves. The body of the slime molds move along decaying twigs and leaves. So here the slime molds engulfing engulfing organic material, right? And under suitable conditions, that means under favorable conditions, slime molds form an aggregation called plasmodium. So here slime molds form an aggregation called as plasmodium. So under suitable conditions, very important for 2 marks question in IB. So here under favorable conditions, slime molds form plasmodium, so which may grow and spread over several feet. So during unfavorable conditions, during unsuitable conditions or unfavorable conditions, this plasmodium differentiates and forms fruiting bodies. This plasmodium differentiates and form fruiting bodies. So these are all the fruiting bodies. And these fruiting bodies bearing spores at their tips. So these fruiting bodies bears spores at their tips. Spores. And here the spores possess true walls. The spores possess true walls. So due to the presence of true walls, the slime molds can survive in extreme conditions. So the spores possess true walls. So true walls are present in the spores. So due to the presence of spore walls, they are extremely resistant and survive for many years, even under adverse conditions or extreme conditions, even under adverse conditions. That means unsuitable conditions. The spores are dispersed by air currents. The spores are dispersed by air current. So slime molds are the saprophytic protistans. They move along the decaying twig and leaves, engulf the organic material, and under favorable condition, they form an aggregation called as plasmodium. Under uh, under unfavorable conditions, plasmodium differentiates into fruiting bodies. So these fruiting bodies possess spores at their tips, and they possess true walls in the spores. So due to the presence of that true walls, they can survive for many years under adverse conditions, and the spores are dispersed by air currents. So this is about the slime molds. So it is 2 marks question in this chapter, right? And also it is important for MSET as well as NEET point of view. So remember the points, that is under favorable conditions, under unfavorable conditions. So here under favorable conditions, it forms plasmodium, it grows over several feet. And under unfavorable conditions, plasmodium differentiates into fruiting bodies. They possess spores. Spores contains true walls. Due to the presence of true walls, it can survive in extreme conditions. So the spores are dispersed by air currents. Right. Next, let us talk about the protozoans. So all the protozoans are heterotrophs. All the protozoans are heterotrophs and live as predators or parasites. They live as predators as or parasites. So they are believed to be primitive relatives of animals. Protozoans are believed to be primitive relatives of animals. Very important bit for objective. So protozoans are the primitive relatives of animals. So there are four major groups of protozoans. There are four major groups of protozoans. Now let us discuss about the four major groups, that is amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. Amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. So this is very important for objective point of view, right? So here amoeboid protozoans. So amoeboid protozoans, these are organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia. So very important, how they capture their prey. So here this amoeboid protozoans capture their prey. So here prey means nothing but food. So they prey by putting out pseudopodia. So pseudopodia is also known as false feet, as in amoeba. And another important thing is so these marine forms have silica shells. So we come across the word silica in chrysophytes and now in amoeboid protozoans. Remember, very very important. So the marine forms, amoeboid marine forms, have silica shells in their surface. Some of them, such as entamoeba or parasites. Entamoeba or parasites. Next, another one, flagellated protozoans. Flagellated protozoans. So here, the members of this group are either free living or parasites, and they have flagella. So the parasitic forms can cause the diseases. Very important. The parasitic form can cause the diseases. The name of the disease is the sleeping sickness. Sleeping sickness caused by trypanosoma. Trypanosoma. Other diseases also caused by these flagellated protozoans like leishmaniasis, giardiasis, etc. So the members of this group are either free living or parasitic, and they have flagella. So the most important thing is a parasitic form can cause diseases. The disease name is a sleeping sickness. Example, trypanosoma. Right. Next, ciliated protozoans. Ciliated protozoans. So these are aquatic, actively moving organisms. That is because of the presence of thousands of cilia. Because of presence of thousands of thousands of cilia. Thousands of cilia are present. So they have a cavity, that is gullet. They have a cavity, gullet, and that opens to the outside of the cell surface. So the coordinated movement, very important. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Example, paramecium. So here, so diagram of paramecium is given here, and this is a diagram of paramecium. It possesses thousands of cilia, thousands of cilia. So these are the thousands of cilia are present, and here the shape of the paramecium is a horseshoe shape. Horseshoe shape, right? So this part is known is the cavity or gullet. cavity or gullet. So the coordinated movement of cilia, they work like oars. They work like oars. So due to the coordinated movement of cilia, the food is entered into the cavity, right? So ciliated protozoans, we're talking about the ciliated protozoans. So they have a cavity that opens to the outside of the cell surface. So due to the coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet, example paramecium, right? So in the amoeboid protozoans, they capture food by means of putting out pseudopodia, that is false feet, whereas here the ciliate protozoan paramecium possess a cavity. So due to the coordinated movement of rows of cilia that work like oars, and that causes the water laden with food to be stirred into the gullet, example paramecium, right? And next, sporozoans. So very important, sporozoans. So here sporozoans include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, that is malarial parasite which causes malaria, a disease which has a staggering effect on human population. So sporozoans, they has a staggering effect on human population. So you had already studied this uh, sporozoans in the first year of your zoology, right? So there you studied in detail about the life cycles of Plasmodium vivax in man and mosquito, right? So this is about the protozoans. So we come across the four groups of protozoans, that is amoeboid protozoans, flagellated protozoans, ciliated protozoans. Okay, right. Next, let us start with the kingdom fungi. So here, kingdom fungi includes multicellular eukaryotic organisms, except yeast. So here fungi constitute unique kingdom of the heterotrophic organisms. Heterotrophic organisms. So kingdom fungi contains heterotrophs, and very important, they show a great diversity in morphology and habitat. A great diversity in morphology and habitat. Whereas kingdom Monera, bacteria are the sole members of kingdom Monera. They show great diversity in metabolic diversity shown by the bacteria, and bacteria are simple in structure, complex in behavior, whereas the fungi show a great diversity in morphology and habitat. So this, you must have seen fungi on moist bread and rotten fruits, the common mushrooms you eat, and toadstools are also fungi. So here the common mushrooms and toadstools, they are nothing but they are edible. And here white spots seen on mustard leaves are due to parasitic fungi. Parasitic fungi. So name of the parasitic fungi is the albugo, which is a obligate parasite, that is albugo. So white spots on the mustard leaves caused by the parasitic fungi, that is albugo, which belongs to the class of Phycomycetes, right? And some unicellular fungi, example yeast are used to make bread and beer. So here, that is name of the yeast is a Saccharomyces cerevisiae. Saccharomyces cerevisiae. Yeast. Saccharomyces cerevisiae. So yeast are used to make bread and beer. So other fungi cause diseases in plants and animals, wheat rust causing puccinia. So wheat rust causing by puccinia. That is also obligate parasitic fungi. So here white spots on mustard leaves caused by the albugo, whereas wheat rust causing puccinia is an example, is an important example. So this puccinia is belongs to the class of Basidiomycetes, right? And some are the source of antibiotics. Antibiotics. Example: penicillin is an antibiotic which is produced from the penicillium, and fungi are cosmopolitan in nature. Fungi are cosmopolitan in nature. Fungi are cosmopolitan in nature. That means they occur, that they distributed in entire world and occur in air, water, soil, and on animals and plants. So they prefer to grow in warm and humid areas. They prefer to grow in warm and humid places. So here from this first paragraph we have 4 marks question, that is advantages of fungi and disadvantages of fungi. So under the advantages of fungi, so you have to write about the uses of the fungi, that is in the preparation of bread and beer, right, as well as production of antibiotics and edible mushrooms and toadstools. And under the disadvantages you have to write the disease names. So white spots on mustard leaves caused by albugo, and wheat rust causing by puccinia, right? So you have to mention about the uses and diseases of fungi in 4 marks question. So in this chapter only four marks questions are there. That is most of the questions from kingdom Protista and one question from this kingdom Fungi. Right. Next, they prefer to grow in warm and humid areas. They prefer to grow in they prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. We often keep the food in the refrigerator to avoid the spoilage of food material. Okay, so with the exception of yeast, which are unicellular, very important. Yeast are unicellular, and remaining fungi are multicellular. So here yeast which are unicellular, fungi are filamentous, and their body consists of long, slender, thread-like structures. So their body consists of long, slender, thread-like structures, which also looks like tube-like structures. And here, so here their body consists of long, slender, thread-like structures called hyphae. So this is known as the hyphae, and the network of hyphae is called as mycelium. So here the network of hyphae is called as mycelium. The network of hyphae is called as mycelium. So this one is a mycelium. Mycelium. So the long tube-like slender tube-like structure is called as hyphae, and it contains some branches. So that is called as a mycelium. That means the network of hyphae is called as mycelium. And here some hyphae are continuous tube filled with a multinucleated cytoplasm. These are called as a cenocytic hyphae. So here, so some hyphae are continuous filled with the multinucleated condition, that is called as the multinucleated, as well as a cytoplasm is also present. Multinucleated cytoplasm is present, and that is called as the cenocytic hyphae. Cenocytic hyphae, right? And others have septae or cross walls in their hyphae. So some fungi possess cross walls in their hyphae like this. So these are the septae or cross walls, right? So here, so this is about the body organization of fungi. So the cell walls of fungi are made up of chitin. Made up of chitin. So here, the cell walls of fungi are composed of chitin and polysaccharides. Very important, chitin and polysaccharides. So most fungi are heterotrophic. Most fungi are heterotrophs and absorb soluble organic matter from dead substrates and hence are called saprophytes. So most of the fungi absorb the soluble organic matter from dead and decaying source. Okay, so and here they are called as uh, saprophytes. Here the dead material is called as detritus. And here those that depend on living plants and animals are called parasites. So some fungi depend on living plants or animals called as parasites, and some show symbiotic association. So symbiotic association with lichens, symbiotic association with lichens. So lichens are the specialized organisms in which algae and fungi live as symbionts. That means here fungi shows symbiotic association with algae. So the fungal component is called as the mycobient, and algal component is called as the phycobient. Phycobient. And here some of the fungi show symbiotic association in the roots of higher plants, that is in pinus. So we had already discussed in the plant kingdom, plant kingdom under the heading of gymnosperms. So here the name of the fungi is the glomus. Glomus. It lives in the roots of a pinus plant, and it helps in the absorption of phosphorus to plants. And this type of association is called as a mycorrhiza. So here they can also live as symbionts in association with algae as lichens, and with the roots of higher plants as mycorrhiza in pinus. Right. So next, reproduction in fungi can take place by means of vegetative reproduction, asexual reproduction, and sexual reproduction. So vegetative reproduction by means of fragmentation, fission, and budding. Asexual reproduction by means of spores, by means of spores, called conidia or sporangiospores or zoospores. So the asexual spores are called the conidia or sporangiospores or zoospores. Conidia, sporangiospores or zoospores. And sexual reproduction by oospores, ascospores, and basidiospores. Sexual reproduction by means of oospores, ascospores, basidiospores. So the various spores are produced in distinct structures called as fruiting bodies. So the sexual cycle involves three stages, very important. The sexual stages or sexual cycle involves the following three steps. The first step is a fusion of protoplasm between two motile or non-motile gametes called plasmogamy. And second one is a fusion of two nuclei called karyogamy. And third one is meiosis in zygote resulting in haploid spores. These three are very important for objective point of view. So the sexual cycle involves three steps. One is the fusion of protoplasm, that is between two motile or non-motile gametes called plasmogamy, and fusion of two nuclei called karyogamy, and meiosis in zygote resulting in haploid spores. Right? So when a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. So when a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. So in some fungi, the fusion of two haploid cells immediately results in diploid cells. So uh, two haploid cells. So fusion of two haploid cells immediately results in diploid cells. That is 2n condition. So very important. However, in other fungi, that is Ascomycetes and Basidiomycetes, an intervening dikaryotic stage, that is n plus n per cell condition, that is two nuclei per cell condition, is observed in Ascomycetes and Basidiomycetes. Ascomycetes and Basidiomycetes. And that stage is called as the dikaryotic stage, and such condition is called as the dikaryon, and the phase is called as the dikaryophase. Very important. Previously, so this statement was asked many times in the NEET examination as well as MSET examination. So dikaryotic condition is observed in which are the following classes, that is Ascomycetes and Basidiomycetes. So in that we observe n plus n per condition, that means two nuclei per cell occurs. So the later what happens? Later the parental nuclei fuse and the cells become diploid. Later the parental nuclei fuse and the cells become diploid. So the fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The fungi form fruiting bodies in which meiosis division or reduction division takes place, that leading to the formation of haploid spores. And very important, the morphology of mycelium and mode of spore formation, the morphology of mycelium, morphology of the mycelium and the mode of spore formation and fruiting bodies, that is morphology of mycelium and mode of spore formation and the fruiting bodies form the basis for the division of the kingdom into various classes. So this is also previous question. On what criteria fungi are divided into different classes? And that is the morphology of mycelium and mode, that is the mode of spore formation and mode of fruiting bodies, that form the basis for the division of the kingdom into various classes. So here four classes are included, that is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Okay, so four classes were included in this fungi. So here, so these are important for objective point of view only. So here Phycomycetes, so here phyco means alga, Mycetes means fungi. So common name is the algal fungi. Common name is the algal fungi. Alright. So here members of Phycomystis are found in aquatic habitat and on decaying wood in moist and damp places or as an obligate parasites on plants. So as obligate parasites on plants. So the mycelium is aseptate and cenocytic. The mycelium is aseptate and cenocytic. In the Phycomystis, the mycelium is, what is mycelium? The network of hyphae is called as mycelium. So the mycelium is aseptate and cenocytic. Aseptate means cross walls are absent. Cross walls are absent. Cenocytic. So multinucleus condition, multinucleus cytoplasmic condition is observed in their body. And asexual reproduction takes place by zoospores motile, or by aplonospores, so which are non-motile. So in the Phycomystis, asexual reproduction takes place by means of motile zoospores or by non-motile aplonospores. So these spores are endogenously produced in sporangium. Endogenously produced on sporangium, and a zygospore is formed by fusion of two gametes. A zygospore is formed by fusion of two gametes, that is by sexual reproduction. So these gametes are similar in morphology, that is isogamous, or dissimilar, anisogamous, or oogamous. So some common examples are mucor, rhizopus, and albugo. Mucor, rhizopus, and albugo. So just to put a mnemonic, and easily you can remember the these examples of Phycomystis. So albugo, mucor, rhizopus are the examples for the Phycomystis. Okay, right. Next, another class is the Ascomystis, and here remember the one mnemonic that. So here endogenously spores are produced endogenously. So here the spores which starts with the letter A and Z, A and Z, that may be ascospores or zoospores or whatever it may be. The spores starts with the letter A and Z that are produced endogenously. You can remember this blindly. Endogenously. Next, Ascomycetes. Ascomycetes. So the common name for Ascomycetes is the sac fungi. Common name for this Ascomycetes is the sac fungi. And here some images are given for Phycomycetes. So the first one is a, this one is a mucor. This one is a mucor, and this one is a aspergillus. So aspergillus niger produces which organic acid? That is citric acid. Citric acid. And this one is a mushroom, edible mushroom. That is agaricus. Agaricus. Right? Ascomycetes. So here Ascomycetes right. So the common name for Ascomycetes is a sac fungi. Ascomycetes. The common name for Ascomycetes is a sac fungi. And except yeast, remaining all are multicellular. So yeast comes under the Ascomycetes, which is a unicellular, unicellular. And other examples for Ascomycetes: Claviceps, Neurospora, Penicillium, right? Aspergillus. So here, mycelium. If you talk about the mycelium in the Phycomyces, mycelium is aseptate and sinocytic, whereas in Ascomycetes, the mycelium is branched and septate. So remaining all the classes except Phycomycetes, that is Ascomycetes, Basidiomycetes, Deuteromycetes, mycelium is branched and septate. Branched and septate. For this, you have to make a table. So comparative studies make you clear the concepts, right? So the asexual spores are conidia produced exogenously on special mycelium called conidiophores. Conidiophores. So conidia are the asexual spores. They are produced exogenously on the special mycelium called conidiophores. So conidia on germination produce mycelium, and sexual spores are called ascospores. Sexual spores are called ascospores, which are produced endogenously. So which are produced endogenously in a sac-like asci, sac-like asci, and these are arranged in different types of fruiting bodies called ascocarps. The fruiting bodies in Ascomycetes are called as a ascocarps. So ascospores are the sexual spores. And examples: Aspergillus, Claviceps, Neurospora, Aspergillus, Claviceps, Neurospora, and Neurospora is used extensively in biochemical and genetic work. So this is a previous question. So Neurospora is used extensively in biochemical and genetic work, right? And here many members like morels and truffles are edible and are considered delicacious, Morels and truffles are edible, right? And another important thing of this Ascomycetes are caprophilous. Caprophilous. That means, so they can grow on dung. They can grow on dung. Dung. And another example for this Ascomycetes is a yeast and penicillium. Yeast and penicillium. Yeast is a unicellular. So remaining all are multicellular. Remember the examples are very, very important. Yeast, penicillium, aspergillus, claviceps, neurospora, morels, truffles, right? And next, Basidiomycetes. So commonly known forms of Basidiomycetes are mushrooms, mushrooms, and very important in Ascomycetes and Basidiomycetes, dikaryotic phase is observed, right? So here mushrooms, so bracket fungi or puffballs. So the common name for Basidiomycetes is the club fungi, club fungi or bracket fungi, club fungi or bracket fungi, and they grow in soil, on logs and tree stumps, and in living plant bodies as parasites. Examples: rusts and smuts, rust and smut. So the mycelium is branched and septate. Just like Ascomycetes, the mycelium is branched and septate. And asexual spores are generally not found. Asexual spores are generally not found. That means asexual reproduction is absent. And vegetable reproduction by fragmentation is common. And sex organs are absent. Sex organs are absent. So here the asexual spores are generally not found, and but so here vegetable reproduction by means of fragmentation common, sex organs are absent. So here plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is called as dikaryotic. As we had studied in the introduction, dikaryotic stage is observed in Ascomycetes and Basidiomycetes. So, which ultimately gives rise to Basidium. And on the Basidium, Basidiospores are formed. So, here karyogamy and meiosis takes place in the Basidium, producing 4 Basidiospores. 4 Basidiospores. These Basidiospores are produced exogenously. Basidiospores are produced exogenously on the basidium, which is a fruiting body. And the basidia are arranged in fruiting bodies called basidiocarps. So here in Ascomyces, the fruiting body is called as ascocarp, whereas in Basidiomyces, the fruiting body is called basidiocarp. So some common examples are Agaricus, Ustilago, Puccinia. Agaricus, Ustilago, Puccinia. Agaricus is a mushroom, Ustilago is a smut, Puccinia is a rust fungi. Right? And next, Deuteromycetes. So Deuteromycetes, the common name for Deuteromycetes is a imperfect fungi. Imperfect fungi. So because only the asexual or vegetative phases of these fungi are known, when the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stages another placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. So that is why it is called as the imperfect fungi. Right? So the Deuteromycetes reproduce only by asexual spores known as conidia, only by means of conidia. And here the mycelium is separate and branched, just like Ascomycetes and Basidiomycetes. And some members are saprophytes or parasites, while large number of them are decomposers of litter and help in mineral cycling. That is, some examples are Alternaria, Coltotrichum, and Trichoderma. Alternaria, Coltotrichum, Trichoderma are the examples of Deuteromycetes. Right. So here another important feature of Basidiomycetes is the presence of clamp connections. Presence of clamp connections in the Basidiomycetes is a characteristic feature of the Basidiomycetes. So this point is not given in our NCERT. So presence of clamp connections is a characteristic feature. Characteristic feature of Basidiomycetes. So you will study in detail about these clamp connections and the classes of fungi in higher class, right? And next come to the kingdom Plantae. Kingdom Plantae. Kingdom Plantae includes all eukaryotic and chlorophyll containing organisms commonly called plants. As we know that they are the autotrophs, and a few members are partially heterotrophs. Very important. A few members are partially heterotroph, such as the insectivorous plants or parasites. So remember the examples. So for 2 marks question, it is very important. So bladderwort and Venus flytrap are examples of insectivorous plants. Examples of insectivorous plants, that is bladderwort and Venus flytrap. And Cuscuta is a parasite. So Cuscuta or Afflicia, Viscum, Striga are the examples of this parasitic plants. So Rafflesia, Viscum and Striga. So these are the examples, some of the examples of parasitic plants, right? So here plant cells have an eukaryotic structure with prominent chloroplasts and cell wall mainly made up of cellulose. Mainly made up of cellulose, right? And here we had already studied in detail about the kingdom Plantae under the chapter of plant kingdom, that is algae, bryophytes, pteridophytes, gymnosperms and angiosperms, right? So life cycle of plants has two distinct phases: diploid sporophytic and haploid gametophytic, that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on others, so vary among different groups in plants. So this phenomenon is called alternation of generation. That is, we had already studied in the sexual reproduction of flowering plant and as well as in the plant kingdom, right? So the cell wall component in the plants is cellulose. Next, kingdom Animalia. So about the kingdom Animalia, you will study it in detail in the zoology part. And next, let us start with the topic virus, viroids, prions, and lichens. Very very important. So for IP point of view as well as a NEET point of view, very important. So in the five kingdom classification, Whittaker, there is no mention of lichens and some acellular organisms like viruses, viroids, prions, and lichens. So these are briefly introduced here. That is, all of us who have suffered the ill effects of common cold or flu, so know what effects viruses can have on us. Even if we do not associate it with our condition, virus did not find a place in classification since they are not considered truly living. Very important. So viruses are considered as non-living. That is why they are not classified under any classification system, right? So if we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Very important. So virus characterized by an inert crystalline structure outside the living cell, right? So here, so two images are given, that is, one is the tobacco mosaic virus and another one is the bacteriophage. Tobacco mosaic virus and bacteriophage, right? So here, so this is the structure of tobacco mosaic virus. So this is a genetic material, and so every virus mainly contain two components. One is a genetic material and another one is a protein code. So the genetic material may be RNA or maybe DNA, and um, genetic material is surrounded by a protein coat called as capsid, and each capsid contains some subunits. So those protein subunits are called as the capsomeres. Capsomeres. So capsomeres are nothing but they are protein subunit. And some viruses contain spike on their body, and those are called as a peplomeres, right? And here, this is the structure of bacteriophage, so which contain head, collar, and tail fibers. So in the head, we'll observe polyhedral symmetry, whereas in the tail, helical symmetry is observed. Helical symmetry is observed in the tail. So polyhedral symmetry, polyhedral symmetry in the head, and helical symmetry in the tail. Helical symmetry in the tail, right? Right. So here, once they infect a once they infect a cell, once they infect a host or cell, they take over the machinery of the host cell to replicate themselves, killing the host. So would you call viruses living or non-living? So now virus is said to be living. Now virus is said to be living. Outside the host, it is non-living, right? And here, so regarding the viruses, so virus means venom or poisonous fluid. And here Dmitry Ivanovsky recognized certain microbes as causal organisms of the mosaic diseases of tobacco, and they were found to be smaller than bacteria because they passed through bacterial proof filters. That is, here Dmitry Ivanovsky wanted to discover or find out the causative uh, organism for tobacco mosaic disease. So for that, he took the leaves of tobacco plant and crushed, and using a bacterial proof filters, he wanted to find out the causative organism, but he failed to find out the that causative organism of tobacco mosaic. And here later on, and that Dmitry Ivanovsky concluded that Dmitry Ivanovsky remember the year also, that is 1892, concluded that some filterable agent is responsible for the tobacco mosaic disease. That means after filtration, he used the sap, and that sap is injected to the tobacco plant. Again, he observed some symptoms, right? So he concluded that some filterable agent is responsible for the tobacco mosaic disease. Later on, another scientist came into scenario, that is Martinus W. Beijerinck, MW Beijerinck, 1898. So demonstrated the extract of the infected plants of tobacco could cause the infection in healthy plants, and named the new pathogen virus and called the fluid as Contagium vivum fluidum. Contagium vivum fluidum. So he called it as, he named the pathogen as virus, and named he called the fluid as Contagium vivum fluidum. And W.M. Stanley showed that virus could be crystallized, and crystals consist of largely of proteins. They are inert outside their specific host cells. So viruses are obligate parasites. Viruses are obligate parasites. Very important. The term is obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. And no virus contains both RNA and DNA. Remember, very important. No virus contains both RNA and DNA. So they may contain RNA or DNA, and they may contain single stranded RNA or double stranded RNA, single stranded DNA or double stranded DNA. So here a virus is a nucleoprotein, and the genetic material is infectious. In general, viruses that infect a plant have single stranded RNA. Very important. Viruses infect viruses that infect plants have ssRNA. Very, very important. And viruses that infect animals have either singular double-stranded RNA or double-stranded DNA. Viruses that infect plants have ssRNA. Viruses which infect animals have either singular double-stranded RNA or double-stranded DNA. Bacterial viruses called as the bacteriophages are usually bacteriophages are usually double-stranded DNA. And here the protein coat is called as capsid. The capsid is made up of small protein subunits called as capsomeres, so which protects the nucleic acid. So these capsomeres are arranged in the helical or polyhedral geometric forms. In tobacco mosaic virus, so 2130 subunits are present. That is 2130 capsomeres are present. So viruses cause diseases like mumps, smallpox, herpes, and influenza. So AIDS in human is also caused by virus, acquired immunodeficiency syndrome. So in plants, so the symptoms can mosaic formation, leaf rolling and curling, yellowing and yellowing and vein clearing, dwarfing and stunted growth. Dwarfing and stunted growth. These are the some symptoms in the plants. So first we'll observe the symptoms in leaf part only. Right? Next, viroids. Very, very important. In 1971, T.O. Diener, it is also 2 marks question. T.O. Diener discovered a new infectious agent that was smaller than viruses. And caused PSTBD, that is potato spindle tuber viroid. Potato spindle tuber viroid. So it was found to be a free RNA and it lacked the protein coat. Whereas in viruses, nucleic acid and protein coat is present. Here, viroids lack the protein coat that is found in viruses. Hence the name viroid. So the RNA of the viroid was low molecular weight. The RNA of the viroid was low molecular weight compared to virus. So virus have high molecular weight of genetic material. And prions. So prions, very, very important for NEET point of view. So in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. Abnormally folded protein, that is the infectious protein particles. They are the infectious protein particles. The agent was similar in size to viruses. The agent was similar in sizes to viruses. So these agents were called prions. Prions. So these are nothing but they are the infectious proteinaceous particles, right? So that means they do not possess nucleic acid. The most notable diseases caused by prions are bovine spongiform encephalopathy, commonly called mad cow disease in cattle, and its analogous variant Creutzfeldt-Jakob disease in humans. So, mad cow disease, also called as BSE, means bovine spongy encephalopathy, commonly called mad cow in cattle, right? And its analogous variant in humans is the CJD. Remember, this question was asked many times in the NEET examination. Very, very important. So, Creutzfeldt-Jakob disease in humans. So, prions also causes scrappy disease in sheeps. Scrappy disease in sheeps. Next, lichens. So, lichens are the symbiotic associations, and that is mutually useful associations between algae and fungi. Algae and fungi. So the algal component of lichens is called as phycobient, and the fungal component of lichens is called as a mycobient. And here algae provides food to the fungi, and in turn fungi provides accommodation or shelter to the algae. And here lichens are good pollution indicators, right? So here, so the algal component is known as phycobient and fungal component as a mycobient, which are autotrophic and heterotrophic. That means phycobient is autotrophic, mycobient is heterotrophic. So algae prepare food for fungi, and fungi provide shelter and absorb mineral and nutrients and water for its partner. So, so close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. సో ఆ లైకెన్స్లో రెండు ఆర్గానిజమ్స్ ఉన్నాయని ఎవరు కూడా అనుకోరు అనమాట. సో లైకెన్స్ ఆర్ వెరీ గుడ్ పొల్యూషన్ ఇండికేటర్స్. దే డు నాట్ గ్రో ఇన్ పొల్యూటెడ్ ఏరియాస్. ఇట్ ఈస్ ఏ నీట్ PYQ క్వశ్చన్. Lichens are the very good pollution indicators and they do not grow in polluted areas.